Active transport uses energy to move solutes against their concentration gradient, not down along the concentration gradient, but against their concentration gradient from lower concentration into higher concentration. Facilitated diffusion is still passive transport because the solute moves down its concentration gradient from higher concentration to lower concentration, and the transport requires no energy. But some transport protein, however, can move solutes against their concentration gradient from lower concentration into higher concentration, spending energy in forms of ATP. That's the active transport. Active transport moves the substances against their concentration gradient. Active transport requires energy, usually in the forms of ATP. Active transport is performed by specific proteins embedded in the membranes. Active transport allows cells to maintain concentration gradient that differ from their surrounding. So maybe outside is very low, but inside the cell they keep very high concentration. The well-known example is sodium-potassium pump is one type of active transport system for animal cell. This one showing exemplary sodium-potassium pumps across the membrane. So for that specific transport protein, for the Na+, they're gonna bind into the something like active site. Yeah. So they bind into the some place of transport protein, and they spending ATP. They change their conformation. Then Na+, they kick out of the cell. Then K+ ion, they bind into the other side of the transport protein. Then it's gonna go into the cell. So get into the cell. So they kick out Na+ and they hold in K+ ion into the cell. By that transport protein spending ATP, that's the one of the example for active transport. So that's why they keep high concentration of K+ ion inside the cell and keep low concentration of Na+ inside the cell compared to outside environment. It's showing you some difference between passive transport and active transport. So passive transport, it is controlled by diffusion. Sometimes just directly pass through that phospholipid bilayer or through that channel protein or carrier protein, but still from higher concentration to low concentration by diffusion. But active transport, they using ATP, biological energy form, and the transport from low concentration is higher concentration against concentration gradient. For the diffusion through the membrane, especially for ions, plus ion, minus ion, we have to add up one more factors to affect that transport. One thing is we call membrane potential. That's the voltage difference across the membrane. Voltage is created by differences in the distribution of positive and negative ion across a membrane. So the membrane inside the outside, usually inside it is charged minus, and outside maybe plus. Then what? That the diffusion of ion, it is affected by that electricity plus and minus. We call that the membrane potential. So before the discussion about membrane potential, we just consider for the concentration gradient of solute. However, we combine together two combined forces collectively called electrochemical gradient. That's the what? Two parts they combine together. So electricity and chemical concentration. That drive the diffusion of ions across a membrane. A chemical force, that's the ion concentration gradient from higher concentration to low concentration for the diffusion. And the other one is an electrical force, the effect of the membrane potential on the ion's movement. So inside the living body, sometimes some cells, they can make those membrane potential on purpose. So some transport protein, they can do those kind of pump. So we call electrogenic pump. That's the transport protein that generate voltage across a membrane. So they making difference, plus charge, minus charge across the membrane. Electrogenic pump. The sodium-potassium pump is the major electrogenic pump on animal cell. They kick out 3+++, and they absorb 2K++. That's why they make a difference. That's one of their kind of electrogenic pump. The main electrogenic pump of plants, fungi, and bacteria is a proton pump. So sometimes they pumping out or pumping in proton plus ion. H plus ion, that's the proton, as I mentioned. H plus ion, that's a proton pump. Electrogenic pumps help store energy that can be used for cellular work. That's why. This one is showing proton pump. the pumping out H plus ion, spending ATP. Another type of transport through the membrane, across the membrane, that's the exocytosis and endocytosis. Exo means to outside, and endo means to inside. So Burke's transport across the plasma membrane occurs by exocytosis and endocytosis. Small molecules and water enter or leave the cell through the phospholipid bilayer or via transport proteins. Large molecules such as polysaccharides and protein cross the membrane in Burke via vesicle. Burke transport requires energy. In exocytosis, transport vesicle migrate to the membrane, fuse with it, and release their contents outside the cell. So like the secretion, as we discussed already before. So contents, packaged with a vesicle, it can secrete out. So that's the exocytosis. Many secretory cells use exocytosis to export their products. In endocytosis, the cell takes in macromolecules by forming vesicle from the plasma membrane. Endocytosis is a reversal of exocytosis involving different proteins. And there are three types of endocytosis. Phagocytosis, phago means eat, and cyto means cell, so cellular eating. Pinocytosis, pino means drink, so that's the cellular drinking. And last one, that's the receptor-mediated endocytosis. This one showing endocytosis. First one, phagocytosis. Second one, pinocytosis. And last one, receptor-mediated endocytosis. Okay, phagocytosis, cellular eating. So, cell can eat up bacterium or food by wrapping up with the plasma membrane, making up food vacuole. And those food vacuole, as we already discussed over, with the lysosome, it can be digested inside the cell. This one is showing you pinocytosis, cellular drinking. Because they, surrounded the liquid part, that's why usually those liquid can be the mixture of many material, many molecules. So it is not very specific they can drink. And for the very specific material they can eat by the very specific cell, then they can go through receptor-mediated endocytosis. In receptor-mediated endocytosis, binding of ligands to receptor triggers vesicle formation. A ligand is any molecule that binds specifically to a receptor site of another molecule. For example, the lipid cholesterol molecule, it is lipid, yeah? That's why when it is transported through the bloodstream, it has to be packaged. It has a coating with phospholipid with a ligand molecule. Those ligand has to be fit with receptor molecule in a very specific cell. So only after binding between ligand and receptor molecule, they have to go through endocytosis. That's why very specific cell, they can go through the endocytosis with very specific molecule. That's why it is very specific for receptor-mediated endocytosis. This one showing receptor-mediated endocytosis, and for some very specific cell, they have a receptor molecule, and those receptor has to make binding with the ligand molecule for the endocytosis. That's the conclusion for the discussion about membrane and membrane transport.